Hey, Mr. P here. In this video, we're going to talk about how meiosis explains Mendel's results. So, in previous videos, we've talked through the processing of meiosis and what the stages are and what happens within the stages. And so, all of that is prior knowledge. We are going to just utilize our understanding of meiosis in this video to understand how Mendel's results work, how he concluded for the results that he concluded, and how we now know, and why we now know, that genes have to be located on chromosomes, which is the model of not only inheritance but genetics. So, in order for us to understand how genes and chromosomes work, is first we need to understand some information about genes and chromosomes. So if we look at this table, genes occur in pairs. We've already established that organisms contain alleles, and by alleles I mean the actual physical letters like big A, little a, or little a, little a. These are individual alleles, and they are contained in pairs. They are inherited as separate units because alleles segregate through the principle of segregation, which we've talked about. Members of a gene pair separate from each other during meiosis. Again, that is called segregation. And members of one gene pair independently assort from each other during meiosis. That's called independent assortment. So in previous videos, we've talked through what the law of probability is, what the law of dominance is, what the principle of segregation is, and what the principle of independent assortment is. All of these are fundamental properties of not only inheritance, but how genes and chromosomes interact to produce the traits that we ultimately express. Chromosomes are going to occur in pairs as well. We call these homologous chromosomes or homologous pairs. We've talked that homologous pairs of chromosomes come together during a process of synapsis. They are not identical. In fact, one of your homologous chromosomes, or homologs, will be paternal and one will be maternal. You get one of the chromosomes of each homologous pair from your dad and one from your mom. Members of a homologous pair separate from each other during meiosis. Okay, that's basically segregation at the chromosome level. And then members of one chromosome pair independently assort from each other during meiosis. Again, that's independent assortment, and that's independent assortment at the chromosome level. So you will see that there are similarities between genes and chromosomes, and they interact not only together, but they kind of mimic each other in terms of how they come together, how they're paired, how they're separated during gamete formation, and then how they are independently transferred from parent to offspring independently of all of the others. In meiosis, members of a chromosome pair separate from each other and members of a gene pair separate from each other during gamete formation. So, we know that through meiosis, we're going to reduce the number of chromosomes and therefore reduce the number of genes from our starting cell, which is diploid, to our haploid cell, or haploid gametes, which again are haploid. And so the way that segregation and independent assortment work within the cells that undergo meiosis is that these chromosomes, which possess the alleles, which are the alternative forms of genes, which are these letters, are going to come together during a process of synapsis, and they're going to independently of each other assort out, utilizing segregation and independent assortment to produce the haploid gametes, which are down here, which are the sex cells in both males and females, which come together during fertilization to produce the physical and genetic components of the offspring of the organism. And so because genes and chromosomes behave in similar ways, genes must be located on chromosomes. That's kind of a conclusion that Mendel came up with, and so that's one of the things that we're kind of focusing on. The fusion of gametes during fertilization restores the diploid number of chromosomes and 2 copies of each gene to the zygote genotype of the next generation. Okay. The chromosome theory of inheritance has been confirmed in many different ways and is one of the foundations of modern genetics. Basically, like I said, the purpose of this video is to kind of not go through what segregation is and what independent assortment is. We've already done that in previous videos. It's just kind of pulling all of this information together and looking at the application of segregation and independent assortment on our way to producing gametes which fuse during fertilization to produce a zygote genotype, which is the actual physical constitution of the letters, which are going to be expressed as physical traits, which is the phenotype. And again, kind of the conclusion or takeaway is that genes and chromosomes behave in similar ways. Again, according to this table, they basically are identical to each other. Genes are in pairs, chromosomes are in pairs. Genes segregate, chromosomes segregate. Genes independently assort, chromosomes independently assort. And so all of this stuff comes together to produce what we now call the chromosomal theory of inheritance. It has been confirmed. It happens this way and has now led to our kind of full understanding, or at least full at this point, understanding of what modern genetics is and what it continues to tell us. Okay, so independent assortment of chromosomes. This is, again, kind of review for what we've already talked about in previous videos, but you know that independent assortment talks about how chromosomes line up, and they line up completely randomly. This is all random. You can have your paternal chromosomes line up on the left and your maternal on the right, or you can have a combination of your paternal on the left and paternal on the right. It doesn't matter. They line up completely randomly and independently of each other, and then based on how they line up, they will assort themselves completely independently, meaning the homologs or the homologous chromosomes will separate, and through independent assortment, you get multiple combinations, which are all genetically unique, based on the sheer fact of how the chromosomes line up dictates ultimately the haploid gamete genetic constitution or the genetic makeup of those gametes through independent assortment and segregation. Okay, the number of possible gamete combinations we always write out as 2 to the nth power, n being the number of chromosomes. And so in this particular case, n equals 2, so 2 to the second would be 4 total chromosomes. Okay, now we know that chromosomes contain genes. The specific site of a gene is called a locus. Plural would be loci. Okay, so this particular chromosome has 2 genes. One gene is the eye color gene, and one gene is the hair color gene. This gene locus is this specific spot on the chromosome that contains the information possessed by that gene. That is the information that codes for the trait, which is the physical expression of the code that is the gene, in this case brown eyes. This would be the second gene locus. Together would be gene loci. It has the physical genetic makeup or the genetic recipe for hair color and will be expressed as the trait, in this case hazel hair, kind of that dirty blonde hair color. Okay. In humans, it is estimated that 20,000 genes are carried on the 24 different chromosomes. Again, we have 22 autosomes, and I should say even further, we have 22 pairs of autosomes. And then we have an X and Y or an X and X. Together, we have 46 chromosomes, okay, in our diploid cells, which again is 2N. Okay? That's all we got for today. Bring your questions to class. See ya.